Hello, and welcome to Conquer RNA Sequence Analytics Challenges with Pivotal and EMC Isilon, sponsored by EMC Isilon. I'm Rebecca Friend, and I'll be moderating this webinar. Our speakers today are Sarah Ernie and Sasha Pagel. You can read their full bios on the right side of your window. Just a few technical notes before we begin. If you have trouble reading a slide, please hold and drag the right corner of the slide window to enlarge. Please also disable your pop-up blocker to participate in the interactive parts of the presentation. At the bottom of your console, you can select the Download Slides button to view a PDF version of the slide deck. And we'll follow the presentations with a Q&A session. Please submit your questions during or after the presentation using the Q&A box at the right side of your window. All right, I think we're ready to begin. Sasha, go ahead.
Okay. Oops, just a sec here. All right. Good morning, everybody. Good afternoon. My name is Sasha Pagel. I'm the Business Development Manager for the Life Sciences vertical here at EMC Isilon. I'm pretty excited to be part of this conversation today. And as the title mentions, we're going to touch on an analysis application called RNA-seq today. But one of the other things that I hope that everybody kind of walks away with is a theme about really sort of shortening the time to results, and this idea about eliminating the movement of data to actually shrink that time to achieve your analysis result. I think, based on this audience here, everybody's probably maybe been tied into the world of scientific research at one point or another, and that desire to get to your results faster is always in the background. So whether you're you're part of the audience that is actually doing the hands-on work like RNA-seq or some of these other methodologies, I hope you walk away with an idea a little bit more about some technologies that are available that will hopefully help you achieve faster or decrease the time to results, and maybe you can bring up with your colleagues that support you with the research IT infrastructure. And if you're part of the research IT group that support your colleagues that are actually doing the science, I hope you also come away with some ideas about some available technologies that you can assist bring to bear and assist your colleagues with, and again, in the spirit of getting to a result quicker. So with that, I'd like to hand it off to my colleague, Sarah, to actually start taking us through the discussion today.
Great. Thanks, Sasha. So, as was mentioned, today I really want to give an overview of how Pivotal can enable work in the field of genomics broadly, and how we can shorten these time to insights in general in the field of bioinformatics in particular, but certainly in genomics today. So at Pivotal, actually to give some background about who we are and what we do, I am part of the data science team, which is a fairly large group of very talented individuals, and actually all the work I'm discussing today will have been performed with a few other individuals, including Marianne McSinai, Noel Seo, Julia Mir-Sarinas, and Ken Dowling, who are all part of the broader data science community at Pivotal. But I really do want to devote some time to actually giving information about what Pivotal is as a company. So it'll become a little bit more clear about how we're actually enabling this type of analysis. So let's start off with sort of the history of Pivotal. So it's a little bit short and long at the same time. We were actually founded earlier this year, and we were sort of assets that were brought together from different parts of EMC and VMware. So essentially all of the big data portions of those companies, and sort of combining that together into one platform so our customers can take full advantage of the data that they have available, doing it sort of application development down to analytics, a very end-to-end solution, giving our customers the ability to really leverage everything that's available. And actually before Pivotal, I was part of the Greenplum team, which is sort of pictured here on the left of this slide. And Greenplum actually began as an MPP database. So I do want to devote some time talking about what MPP databases are and sort of why they're so useful. I'm going to give you a sense of why we are able to accomplish what we have so far in this field. So at the essence of Pivotal Greenplum technology for the database, MPP database, is this concept of being able to take your traditional Postgres-type databases and really expand them to function in a parallel fashion. So what we have here is essentially a way for us to be able to take your data that you would ordinarily store in a table, maybe on a single node, maybe your laptop even, so you can access it and sort of look at it, quickly query it, and really distribute that data across many nodes so that you can, in parallel, search and query that data and actually perform analytics. So in particular, the database itself that we have here at Pivotal has really been focused on optimizing analytics. So essentially, everything that's done, sort of the secret sauce around query optimization, has been heavily focused around how to make analytics fast and how to optimize the queries. So that, very simply put, if you imagine a large table of registered voters in the United States, and you're interested in quickly figuring out the number of people with every name available that exists in this registry, instead of scanning one table, you can sort of split it up and have it automatically be scanned without you actually needing to do much on the front end in terms of thinking about how to send out this query or partitioning the data. It's all sort of done automatically and cleanly for you. Now the real addition here in Pivotal, and that'll be the focus of this entire talk, is that there's been an emphasis around how we actually take advantage of data that can't be stored in a traditional database table. So not everything is what we call structured, in the sense that rows and columns don't necessarily represent this. And actually sequencing data is a good example of something that does not start off inherently structured, but something where you actually add structure to it sort of as you move along. And then in particular, something that we focused on here by starting to think about how we incorporate this external data, in particularly how we bring in Hadoop, which has now become sort of the cutting edge of how you bring in this unstructured data source. And in fact, we started off sort of as thinking about how to integrate that and pull it in a very parallelized fashion sort of in the back end, the way it's depicted here in this image, but actually pushing further into it and releasing our own Hadoop distribution. And I'll go into a little bit more detail in just a few minutes after I pass it over to Sasha about how we've sort of moved forward from that, and also how we do that in sequencing. But I'll let Sasha sort of take it from here.
Great, thanks, Sarah. So just very quickly, I just wanted to just highlight what a traditional sort of Hadoop cluster environment looks like, and what individuals need to do to set it up. And so typically, a Hadoop environment requires something called a name node, which manages all the different block data or data that's managed by the Hadoop distributed file system, metadata. There's also the data node, which actually takes the jobs and distributes them across the different compute nodes. So one of the things that I just wanted to point out very briefly is that typically when you're taking advantage of this environment, when you're bringing in primary data from a variety of different sources, there's actually a number of steps to actually finally to get that data to the Hadoop cluster. So as you can see in this simple slide is that ultimately this data needs to be copied 3 times. Once to the landing zone, from the landing zone actually to the Hadoop cluster, and then eventually that data is then replicated 3 times with each of the different nodes in the cluster. And then only at that point in time do you actually sort of execute your Hadoop analysis. And so that introduces a variety of challenges, whether it's being able to scale this compute, because both the compute and the attached storage are tied directly to each other. So in terms of actual physical assets, you need to scale both at the same time. Or from a different perspective, there's the potential that your job may fail and you can't actually pick it up and continue with your job, which means you have to restart it. So in the context of Isilon, whoops, let me just skip ahead here. Sorry. So for those of that you may or may not know, so Isilon is a scale-out network attached storage, and its operating system 1FS actually provides a variety of support for a variety of different protocols, including HDFS, which is the Hadoop distributed file system. And as such, that gives us some ability to actually eliminate the series of the actual steps of copying the data multiple times over to the Hadoop cluster. Essentially, the compute resources that you have available to you for Hadoop can be pointed directly at the Isilon cluster, and the Isilon cluster can be treated as the named node. And so, in addition to eliminating the steps of copying, you get other benefits like being able to scale out your compute independent of the required storage, or from a different perspective, you can actually simplify bringing in data from a variety of different sources, whether it's through NFS or HTTP or even HDFS. That whole process is simplified by the support for multiple protocols. So the key point here was just that now we're eliminating that entire movement of data earlier that we saw before with a traditional cluster. So I'm going to pass this back to Sarah to move us right along.
Great, thanks. So one additional piece to add to the story around Hadoop and Pivotal is that recently Hawk was released, which is Hadoop with Query. And this is really the concept of taking this database that we have, the MPP database, where we have a nice SQL interface that allows you to access your data, query it, take advantage of the query optimization that's analytics focused, and translate that into HDFS. So what essentially has been done is taking that MPP database and sticking it on top of HDFS. So now you can query things that are in Hadoop sort of the same way that you would normally have done if it were sort of in a traditional database storage. And so the reason for all of this, for the MPP database that is analytics-oriented for Hadoop and general MapReduce jobs, and then now as well the Hawk portion, is that we are trying to eliminate exactly what Sasha said, the need to move data around to truly shorten time to insight. And the reason for this is we understand that data is large, and not only that, data is growing. So as things expand, it no longer is feasible to be moving around data to the different places where you generally compute. In fact, what you want to do is take the applications that are doing that compute and bring them over to where that data lives. So why do we want to do that? Well, let's just think about this in terms of what we would sort of traditionally be doing. What we see a lot are cases where large amounts of data are stored in certain locations that could be in a database or in lots of different file storage systems that could be distributed. I know when I was in grad school, I would have files all over different systems. And essentially what I would want to do is bring those pieces together and start trying to build some sort of an analytics tool around that. So I might be wanting to do something predictive. For example, let's take hospital readmissions. I'm wanting to understand whether or not a patient, given a lot of data that I have, would need to be readmitted following, for example, a surgical procedure. In order to do something like this, I would take a sample and sort of build my in-memory statistical analysis. So I use a tool like R in order to build a predictive model. And that prediction I would then want to use in some sort of a case to try and optimize a decision about whether or not I want to keep a patient in-house. And what I would then want to do is sort of have this general solution that I would plug in and sort of analyze in the larger context of a large volume of data and have to iterate. And really what this means is in learning how this particular process would apply to my large amount of data that's available, it would be a very slow, painful process in order to get sort of moving and iterating on this concept. So really what we want to do is have an integrative architecture that allows us to understand and quickly move around with data. So get a better picture of what's available to us and how we're going to want to compute on all of those items. Really, the holy grail that we're looking for is in the field of biomedicine, the 360-degree view of a patient. If I can understand all of the pieces that make up a particular patient and their condition, his or her condition, then I should be able to build a better model about whether or not I think they will contract a certain disease or be readmitted to the hospital. Now the challenge is that data comes in all shapes and sizes, And there's no sort of one-fits-all solution to this. Just like we've discussed, there are structured data sources like your medical history, which could be your diagnosis codes, medication lists, prior lab tests. These can be large volumes of data that, of course, fit nicely into an MPP database, and we would be able to analyze those nicely sort of in our MPP database structure. But there are a lot of things that are actually unstructured data, like genomics data, that has to be extracted, and you might want to be identifying the variants or the gene expression levels. You might have imaging data that in general is boiled down to some number or score, but in actuality there are a lot more features that you might want to extract from a particular image. And so in fact what we want to do is have a landing zone for all of this data, so that ultimately what we can do is grab features out of those. So create structure around this very unstructured data so we can bring it all together and get really this integrated phenotype that's giving us all of the information that we can then use to create, for example, more targeted treatment, where we are integrating all available data, the medical history, the clinician's notes, the genetics, and the imaging to push and improve our model fit. Now, the reason why Pivotal is ideal for this is really what we've discussed so far, that we can bring together all of these different data sources. It's a landing zone for sort of everything. And I do sort of want to go through an example of what an actual MapReduce job involves. Just in case there are some people in the audience that haven't had the opportunity to work with Hadoop before. And in general, the easiest way to think about it actually is with text data, but of course it can be easily thought of in the context of genomics, and we'll cover that as well, of course. So at the baseline, what we can imagine is working with a bunch of text files. Let's say this is notes on a particular patient. And what we want to do is in this very unstructured data, we want to extract things that are informative, things that we can use for features for building a model. For example, that could be patient history in terms of diagnoses, procedures that they've undergone, or particular drugs that they might have taken. So what we want to do is go through this unstructured data, a bunch of text, and identify, for example, controlled terminologies of interest. And we can do this by sort of processing it one by one the way we would traditionally imagine doing it, or moving the data into clusters. But really, that's not how we want to do it. We want to be able to analyze that data where it lives. And this is why, in general, using HDFS and sort of the MapReduce framework, what we can do is have nodes that are each housing different data. So they have a collection of different sets of notes for different patients, for example, and each one of them can individually in place compute and identify the number of controlled terminologies available in any particular document in a map phase. And in a reduce phase, what happens is all of those things are aggregated onto single notes. And so, for example, you could be aggregating this on a per-patient basis. And what in the end you obtain is a very structured set of data where you've extracted features very quickly without having to move the data around. So the benefit, of course, of having the Pivotal environment is now you can use either Hawk or our MPP database with the connectivity to Hadoop to be able to now integrate that data with other pieces that are already structured. To bring another example forward would be to use image data, where again we have a lot of different features and things that we want to compute, again large volumes that don't need to be moved around. But you can create these different featurizations, for example looking at density and different parts of the image or identifying nodules, aggregating that, and for example across disease classes get a sense of how this information differs in order to build better models. Now we're actually here to talk about genomics. And so let's think about sort of the general framework of how that might apply in the field of RNA sequencing, for example. So a very simple view of that would be that the map phase would actually be looking at all of the raw reads and mapping those to the genome. So in fact, you could look through and scan every single particular read that's available and map it to a different region of the genome, and then you want to aggregate it, for example, at the gene level to be able to get account for the number of reads mapping to that particular gene that are available. And you could aggregate that across patients and, again, compute many things in parallel. So we did have a project where we worked on this, and of course we're not in the business of reinventing the wheel. In fact, what we want to do is leverage what's available there. And it turns out earlier this year there was a fantastic publication sort of summarizing all of the available tools that have been adapted or in some way have been made available in the MapReduce framework. And so, in fact, there are now ways of taking that data and applying sort of your standard tools, so taking, for example, Bowtie, and applying that to your data that's living in HDFS. And so really what you've done is taken what is traditionally done outside of a place where all of your data is stored, taking your raw reads and processing them, for example, outside, and then bringing all of that mapped information in, where you might be wanting to combine it. And we see instances where people are bringing this forward into a data warehouse, running everything separately and outside, and then having to go through iterative steps where, again, they're using just a central place to store the data in order to access it. What we're really trying to advocate here is taking that data and putting it all inside of the Pivotal environment. So again, in addition to actually running your read mapping sort of in our Hadoop framework, we also want you to be able to throw in your image data, throw in all of your external data that's relevant. And again, eliminate that data movement, but also enable the rapid reprocessing of the data in context of other data that's available and integrating it with all of those other data sources. So you might be thinking right now, well, you have a certain set of data that you're operating with in some ways, but ultimately that data can be integrated with a lot of things that are available. So let me just kind of pause and step back and give a different visual of what we mean by all of this. So we went through, at a very, very high level, what in particular would be done for read mapping, for example, in a Hadoop framework. So what we're saying is that in the Pivotal environment, again, you can be running your read mapping sort of as a step and not moving it at all, but in fact just computing sort of in place. Now what you can do is have several things, for example, variant calls or read counts that could come out of that. And in fact, we've toyed with a lot of different options here as to what you can do with the data that comes available. So there are a lot of things that might be of interest. You might be wanting to look at a particular individual's genome, look at mutations, see how they fall into different pathways. And again, having all of this data available in one location really allows you to query the data and to play around with it, go back and look at different pieces of it. Now, another huge benefit, of course, is this concept of integration, which we've really been talking about heavily. And for that, we sort of wanted to step back and walk through one example of how enabling sort of compute without data movement is important by going through, I think the simplest example, the most straightforward one, the genome-wide association study. So let's take the case of having called variants that you've taken in, they live in your database, and you are now trying to understand in the context of all other available data, which of these variants is highly associated with a particular phenotype or a disease. Now, at the beginning we were talking about what you can do in terms of an MPP database to store large volumes of data and be able to run through it and look at what's happening quickly. There seems to be a bit of a formatting issue here, but in general what you can envision is you have a set of n individuals, and for them you have a few covariates. That could be the sex of the individual, their height, their weight, and then you have their SNP data for up to m SNPs, for example. And sort of the way you would think about that is as a sort of Y table where every individual is listed. Now, we can distribute all that data, and of course we can do some very nice computations that way, but the one thing that's very important when you want to bring this data into the database and to really take advantage of the fact that we can rapidly compute and store in one location is how, in that paradigm shift, you'll have to think a little bit differently about how you're going to store the data. And beyond that, everything happens automatically. So in fact, what you'll want to do is take the genotypes and store them in a different way, sort of in a longer table, where you have entries that have individuals, the particular SNP of interest, and the genotype. So for individual 1, SNP, we'll just call it SNP1, has genotype AA. What you can do is actually have all of SNP1 for all of your N individuals living on one single node. And in fact, SNP2 could live on another node. And in the sense that you can distribute those by SNP across the various nodes, you can now very quickly collect, for example, summary statistics about the number of individuals that have any particular genotype. That'll let you compute things like minor allele frequencies in parallel and very rapidly, let you query the data in different ways, and bring it together again in the context of other covariates that are of interest to you. Now, in addition to being able to do those types of analytics where you're getting summary statistics, our platform also allows you to use what's called a procedural language. And so in fact, if you are doing some kind of an association study where you have a statistical test that you're performing in what's called an embarrassingly parallel fashion, where you're generally taking that particular test and applying it to every single one of your genotypes in turn, you can actually, again, run that in parallel by having every segment have the R code registered. And so essentially it just becomes a simple query that you're running, and it automatically runs that in parallel on every separate node. It's actually quite simple. You just take your code that you're normally running, and often people are doing this on clusters by moving the data over. And in fact, you're just allowing the data where it lives to take advantage of the parallelism and simply run that particular R code on that instance where it's living. Finally, you might be interested in some other pieces like the odds ratios. And so what we wanted to do was put together a visual about how you could do that type of computation. And so we took the HapMap data, which of course is artificial, and sort of created a phenotype around the different ethnicities, and again showed how by adding a little bit of noise, you could do this rapidly in parallel and quickly manipulate the types of data that comes out of it. So I'm going to skip this slide in the interest of time and talk about how you would then visualize it. So what we did was you can take any one of your regular tests that you would perform in order to assess the significance of a particular SNP. And we did just that. We took the HapMap data, added a little bit of extra number of individuals, a little bit of extra noise, and computed the significance of being associated with a particular ethnicity across those different SNPs and added some covariates to that. So a couple of benefits, aside from being able to do this extremely, extremely rapidly in parallel without any data movement, is that you can also visualize it very quickly by using tools that have been built for visualization in MPP databases. And so here we used one visualization tool, Tableau, and simply plotted the p-values, or the negative log of those, across the different chromosomes and loci. And here we just show 3 chromosomes for simplicity's sake. So one of the benefits is, of course, this very quick visual that you can have, but these tools are also excellent in that you can link other data to it. And so in particular, what we did was actually include information about the covariates. And so on the bottom left here, you can actually see in Tableau, you can highlight a set of SNPs, which we've done here, and you can link the information so that now you're actually querying another table that's also existing in this database. And again, rapidly and in parallel, it's going to create counts and distributions and tell you how many individuals of each ethnicity and what the different covariates are for the different groups by SNP, grouped by SNP, and the particular alleles. In addition, we added the odds ratios and showing the distribution of those as well in the right bottom-hand panel, again for those selected SNPs. Now, sort of a final benefit of what we wanted to add to that was to be able to say, well, if you actually did have some kind of actual sequence data, and often we see people come in and saying that they do often need to go back to the raw reads to be able to validate whether or not what they're observing is true signal. You can, in fact, highlight a few of those SNPs, and what we did was take a publicly available genome that had been sequenced and allow us to point and essentially look at all of the reads that map or that contain that particular SNP. And here we've just posted up the average map quality and the counts of the number of reads that contain that particular SNP. And so again, by using this MPP technology, these types of searches are enabled and made very rapid. I just want to throw this slide up here really quickly because in addition to what we described here in terms of being able to actually apply things directly, especially in things that can be done in a very parallelized fashion by running many different similar tests or queries that are grouped by individual SNPs, there are things where you might want to actually aggregate all of the information. And so we've actually created at Pivotal in collaboration with a few academic Berkeley institutions, Berkeley, Wisconsin, and University of Florida, an in-database and also in-hoc machine learning library that allows you to take all of this data and actually build one enormous model on data that would normally not fit into memory. So I know this is very short, and hopefully I've succeeded at least in piquing your interest in getting you a sense of what is possible in this type of environment, and how hopefully we are going to be able to enable your research groups to move forward. So I'll pass it back to Rebecca at this point.
Okay, great. I think it's time to move on to Q&A. And we've got a lot of great questions, and we'll try to get to as many as possible. I know we're running a little bit short on time, but we'll still try to get to as many as we can. This first question, I think, is for Sasha. Which version of HDFS is supported by Isilon?
Yep, good question. So actually, both the original HDFS.1 release and then now actually more recently we also support HDFS 2.0. And then probably with that, there might be the question where, along with that, what sort of Hadoop distributions does Isilon support? And of course, Pivotal. There's also, the primary Apache and Cloudera and Hortonworks, for example. So we support a wide variety of Hadoop environments.
All right, great. I think we'll try to get one last question in before we close up. I think this one would go to Sarah. And this is, do I need to engage with Pivotal Labs to get access to and use the Pivotal technology stack?
Oh, so actually it is not required to get in touch with Pivotal Labs directly. I am part of Pivotal Data Science team. We have Pivotal Data Science Labs. Pivotal Labs would be the group that you turn to for rapid application development if you're trying to build an application around something that you're doing with your data or in general an application that you'd like developed. But the Pivotal stack is available, and actually there are sort of single node editions that you can download if you just want to play around with the database, for example. And for example, if you're interested in MadLib, MadLib is an open source library that's available at madlib.net. But certainly different technologies are available in many different ways. And I encourage you to go to gopivotal.com to explore what's available.
All right, great. Unfortunately, I think that's going to be our last question for the day. But I want to thank everyone for attending this Fierce Live webinar and submitting so many great questions. We can also get back to anyone that submitted questions personally after the webinar. I'd like to thank our speakers for participating and EMC Isilon for sponsoring today's webinar. The webinar has been recorded, and you'll be able to access the recording again within 24 hours on the same page that you used to register for the event. Thanks again for joining, and we look forward to seeing you at future events.